Well, thank you very much for the invitation to present this talk on the immunological perspective. I do hope everybody has some coffee left out there because we are going to talk some pretty serious immunology. But it's an exciting time because I'm going to show you a lot of studies here that are really only published in the last 2 years. And I'm going to go through the slides pretty quickly because there's so much to cover, but we actually can find most of these references in a recent review that Antonio Bertoletti and I published. So to get started, in terms of an immunological perspective, we need to understand what you need for immunological control of hepatitis B virus. And to be honest, in general, this is not much different than you see in any other normal acute viral infection. You need T cells that can recognize an infected target, and most importantly, produce cytokines, antiviral cytokines like interferon gamma, which is actually responsible for a majority of the non-cytopathic clearance of the virus. And then, of course, you have antibodies that neutralize the virus. And the big difference here is it takes 2 to 3 months to clear acute hep B versus another acute viral infection. So when you look in the context of a chronic infection, you don't see this multi-specific T cell response effect. It's very weak. We have very few antibodies or ineffective antibody responses, and the T cells, if they are present, are actually prone to apoptosis, or they co-express a number of inhibitory receptors like PD-1, CTLA-4, and TIM-3. So this is actually quite easy to quantify experimentally as well. So if we do an ELISpot, for instance, using overlapping peptides covering the hepatitis B genome, and you look in a resolved patient, each one of these black bars represents a T cell response in a resolved donor and a resolved patient. You look at this same response in a chronically infected patient, and about 80% of the time ex vivo, this is what you'll see, is virtually non-existent. So I think there's 2 very broad and very general reasons that you see this level of exhaustion of this immune suppression in chronic hepatitis B. One is the fact that it's a hepatotropic virus, and we're quite aware that the liver is generally a very tolerizing organ. You have high IL-10, TGF-beta, PD-L1 enzymes that degrade arginase and tryptophan, which are all very suppressive for T cell responses. Coupled with low MHC on the hepatocytes as well as suppressive dendritic cells, makes it difficult under resting conditions to stimulate an effective immune response. Coupled with this hepatotropic infection is the high antigen burden that we see in the hepatitis B patients. And this is one of the things that fascinate me the most probably about the virus, is that it's incredibly small, makes only 4 proteins, but its synthetic effort is really phenomenal. I mean, the level of viremia in these patients is often 10 to the 9, can be 10 to the 9, 10 to the 10, and I've worked with some of these patients where in their serum we can see up to 1 milligram per mil of viral antigens. So there's really a massive antigen burden in these patients. So then the question in terms of immunology is, how can you actually restore a virus-specific T cell response that's been exhausted from years of exposure to an inhibitory environment like the liver, and coupled with the amount of antigen that's present in the circulation or in the body? So your first thought, obviously, is therapeutic vaccine, or at least for an immunologist, is therapeutic vaccines. And Dr. Janssen attested this before. We've been trying now for at least 15 years, and I could go through each of these individually, such as recombinant antigen-based vaccines like the prophylactic vaccines, or going and adding a TLR agonist to immunodominant peptides, DNA vaccination, or immune complexes with alum. But the ultimate response is that after vaccine therapy, we see a transient response. There might be a little bit of a boost in the T cell proliferative response. There might be a slight reduction in the viral DNA for a little bit, but ultimately there's not a durable response. You don't get any good CD8 T cells, which are really responsible for the clearance. And there's very little S antigen loss. So there has been some advancement, though, in the vaccine field, and so in a lot of this, and I'm going to come back another study later, is through the Roggendorf group using their chronic woodchuck hepatitis model, and a new approach, or the most updated approach for inducing T cell vaccines, this combination of antiviral therapy with a prime boost regimen that incorporates adenoviral vectors with a DNA prime boost strategy, and this has been extremely effective in animal models like the mouse and the woodchuck at really inducing a good T cell response. The caveat to this is they have to hit these animals with many, many doses of the vaccine. So you can see it's hard to read actually, I'm sorry about that, but it's 3 doses of a DNA vaccine alternated by 4 doses of an adenoviral vaccine, and then coming back with 2 DNA vaccines, all in the context of antiviral therapy. So giving this kind of aggressive regimen, you do start to see this restoration of a CD4 T cell response about halfway through the vaccine regimen. But in fact, the CD8 T cell responses are much more delayed. So they come up here at about the end after you give the last 2 boosts of DNA vaccine. So what does this look like in terms of a virological response or an immune response to clear the virus from these chronically infected woodchucks? Well, in this woodchuck, it looks fantastic. So you start off at the beginning of therapy, entecavir starts to reduce the viral load, and upon starting the immunization schedule, you see a reduction, almost an elimination of S antigen, and then the woodchucks become anti-S positive. So the caveat, though, to this one is that the S antigen here in this woodchuck is extraordinarily low. It's 5 micrograms per mil. And when they actually looked at the other animals in this group, there was only 4 in the whole study, but when they looked at the other animals that had a much higher S antigen, you can see that it does come down and DNA goes away, but after stopping and after the end of the vaccine therapy, even this very aggressive approach is not necessarily able to end up with stable control over the virus. So it does look like there is some benefit because you get the reduction in the set point for the S antigen, but it's still not be able to clear. And I'm going to give you my hypothesis as to why this is probably the case. You may not agree, but I think this is the case because when you talk about vaccines, you're talking about a needle, a shot in the deltoid or in the arm, and you're giving a very local injection. So in the case of recombinant antigens, a lot of times it's 10 to 50 micrograms of antigen. If you come back to what I said in the introduction about the synthetic effort of the virus, they have high viral load, and it's not unusual to see a patient with 20,000 units of S antigen in the serum. So if you just do some quick numbers based on this, you have 5,000 milliliters of blood, 100 micrograms per mil of antigen in the blood. That's 500 milligrams of S antigen in the patient. So a vaccine with 50 micrograms is equal to half a milliliter of blood. Why should the immune system look at what's going on in the shoulder when it's overwhelmed with antigen in the rest of the body? So that presents an even bigger question. How do you actually deliver a stimulation to break this virus-specific T cell exhaustion? And so what I think is you really have to think on a larger scale and reshape the entire immunological environment. Think systemic versus the local injection of the vaccine, and try to exploit the natural immune mechanisms and also take advantage of the viral life cycle, because the virus is giving us plenty of antigen to use. There's no reason we have to put more antigen in to get an immune response. Plus, the antigen you're putting in is not matched to the virus that's in the patient, so you have issues of viral diversity. So how do we do that? Well, I think there's actually hints from some of those studies I showed you in the previous 2 slides. So this vaccine study with the immune complexes in alum, the responses in this group were to the vaccine were no better than alum alone. But if you look, I don't know if you can see it, but these patients with alum alone, 20% of them seroconverted E antigen. So even the adjuvant alone had an effect here. It was actually a little bit better than the vaccine, which was 14%. And then here in this DNA adenoviral prime boost study from the Roggendorf lab, in their woodchuck hepatitis virus mouse model, they were hitting this mouse with a core antigen vaccine over and over, but then they saw a boost in the anti-S antibodies, suggesting that this kind of adjuvant effect has could have some kind of immunological effect overall, not specific to the vaccine. So then one of the things I'm particularly interested in, and a lot of studies are coming out to support this, is can an adjuvant or an immunotherapy actually rescue the HPV-specific T cell responses? And in terms of adjuvant therapy, I think the one that we're probably all most familiar with right now is this the Gilead compound, this GS-9620. And the concept here is a TLR7 agonist, and the original idea is it stimulates interferon alpha production, which then goes on to have its antiviral effect. And so the good thing about this is, TLR7 is very immunostimulatory. Maybe the problem with interferon alpha is we know it's not necessarily curative. There's good studies now to show that it does not restore T cell responses, and then obviously it has its side effects. But interferon alpha is obviously not the only cytokine that's going to be produced in response to TLR7, so there might still be some adjuvant effects we haven't really seen, or at least I haven't seen data on the T cell responses of HPV-specific T cell responses so far after administration of this compound. And then we published a study just this year looking at TLR8 specifically, and TLR8 stimulation of intrahepatic monocytes. And what we found was that when we stimulated intrahepatic monocytes with this TLR8 agonist, they produced loads of IL-12 and IL-18, and this worked on NK cells and mate cells, this mucosal-associated invariant T cells. This is an NK T cell in the human liver, and it stimulated these cells to produce massive amounts of interferon gamma. And these two lymphocyte subsets probably compose 40 to 50% of your intrahepatic lymphocytes. Interferon gamma is critical for the non-cytopathic clearance of hepatitis B. So, using the right TLR adjuvants, you might get the right antiviral cytokines to get hepatitis B clearance. This was not done in the context of hepatitis B, but it's an extrapolation from the data. And then there was another study just published last year from Percy Nola's lab looking also at the activation of intrahepatic monocytes, but in this case, in the context of TLR9 stimulation. And when they stimulated the intrahepatic monocytes with TLR9, they found that they produced TNF-alpha and recruited T cells to the liver. And they formed these aggregates, and these aggregates facilitated intrahepatic T cell proliferation. And so you got this massive boost in the HBV, or in the antigen-specific T cells in the liver. And so when they combined this TLR9 immunotherapy with a DNA vaccination in chronic hepatitis B infected mice, what they found was that there was a much better response in response to this combination vaccine and adjuvant therapy, or vaccine and we'll call it adjuvant therapy, compared to either the adjuvant alone or the vaccine alone. So suggesting that this modulation is having a beneficial effect as well. But there's also immunotherapy, and this is slightly different because those were TLR agonists stimulating antiviral inflammatory cytokines. But there was also a paper published just last year from Masanori Isagawa from Frank Jazari's group at the time, where they targeted myeloid dendritic cells, these cells that are very important for presenting antigen and initiating the immune response. And instead of giving them a TLR agonist, what they did was target a co-stimulatory molecule, CD40, on these dendritic cells, which upregulates their ability to process and present antigen. And in their HBV transgenic mice model, when they gave them T cells without this adjuvant therapy or without this immunotherapy, the T cell response was weak, hardly ever produced any cytokines at all. However, when they gave this anti-CD40 therapy, you can see this massive boost in the CD8 T cells in the liver. A lot of these cells produced a lot more interferon gamma, and you can see in the blot over here that they almost completely eliminated HBV DNA from the liver in the context of this adjuvant therapy. And so another thing that we've been interested in is along these lines as well, thinking about the antigen burden in the serum of the patients. We wanted to see if any of the monocytes of the dendritic cells in the patients were actually internalizing and using this antigen. And what we found was that this classical population of monocytes, the CD14 monocytes in the blood of hepatitis patients, were loaded with the S antigen. And these are consequently the cells that become dendritic cells and present antigen, and they are the same cells that we use for DC vaccines. And so we simply asked, if we converted these monocytes to dendritic cells, can they present that antigen that they accumulated in the circulation of the patients? And seen here, we saw in 8 out of the 9 patients, this was the case. And what we did, we went on to differentiate these cells to dendritic cells, mix them back with their autologous PBMCs, and saw expansion of autologous T cells, suggesting that there is a therapeutic effect. So that's well and good if there's a T cell response to boost, but I think in many cases, these T cell responses can even be deleted in this really, really high antigen load patients. And so what do we do then? And in that case, there's also an arm of research going on that is much more experimental in nature, but you make a T cell response. You engineer your T cell response that you're interested in. And one way to do that is through a chimeric antigen receptor. And basically, this is an antibody that you modify, clone the DNA, put it into a retroviral vector, and then drop it into the T cells of a chronically infected patient. It's easy to detect this, so when you put it in, now you have T cells that will recognize the viral protein, in this case, the surface antigen. And these T cells will recognize the surface antigen on the surface of hepatocytes, and when they put them into HBV transgenic mice, they saw that these modified T cells reduced the HBV load in the transgenic mice. So this is another thing that we've been working on as well, but also in a slightly different context, in T cell receptor gene therapy. And what T cell receptor gene therapy is Here we actually identify T cells from resolved hepatitis B patients that we know recognize infected hepatocytes and can kill them. And so we clone the DNA that encodes for that receptor, and then it's the same process. You put it into a viral vector, and then you introduce it into the lymphocytes of a chronic hepatitis B patient. And you can see very clearly, if you don't introduce the receptor, there's no positive population that are HBV specific. If you do, we see a nice population of T cells ready to respond to hepatitis B infected cells. And we showed that these cells were functional in an in vivo mouse model where we put a tumor in, let the tumor grow, and then we transferred in these genetically modified T cells. In the control, you can see the tumors here, and then a dose-dependent reduction in the tumor volume in these mice. So suggesting that these T cells can target tumors expressing HBV antigens, which is an important concept because this happens a lot of time in HBV-related HCC. And in fact, through a fantastic collaboration with colleagues in London and colleagues in Pisa, we're able to use this exact therapy in a compassionate care use basis on a patient that developed HBV-related HCC. So just go quickly through the medical history here. And so the patient was diagnosed with chronic hepatitis B, developed liver cirrhosis, and then 2001 developed HCC, underwent transplant, was fine for 5 years, and then detected HBs antigen in the circulation. So after that, they found that the patient had developed metastases, but these metastases were actually from the original liver. They weren't from the new transplanted liver. And so they tried to surgically treat this, but ultimately we got compassionate care use, the possibility to use our genetically modified T cells, because what we could do with our T cells is we could make T cells that specifically recognize the metastases, but not the liver of the patient. So minimizes any risk to causing liver failure in this case. And so we did this, and so this is a history of the S antigen here from the patient, looking from 2011 up till 2012. You can see a dip upon surgery and resection, but it kind of gradually increases. And at this point, we infused the genetically modified T cells, and within 30 days, this went from about 3,500 units per mil down to about 500 units per mil of the S antigen. And so this drop in S antigen correlated with an increase in the frequency of our T cells in the peripheral blood of this patient, as well as a slight increase in ALT, presumably coming from these tumor cells that are being lysed. So this is, unfortunately, it wasn't curative in this case, and the patient did succumb to a cerebral metastases, but there was no adverse effects from this adoptive cell therapy. And I think these are encouraging results, even though it is one compassionate care use basis. But the fact that we did see some biochemical evidence that these cells were effective. So just going to wrap up quickly, and unfortunately I didn't get to put everything in here because, and some people have talked about these use of checkpoint blockades in previous talks, and I think there is a lot of potential here, especially, there's a lot of great in vitro data using the blood from chronically hepatitis B virus infected patients. And then back again from the Rogendorf lab, they did this vaccination protocol with chronically infected woodchucks, either with combination therapy or combination therapy plus anti-PD-L1 blocking. And in the blue line here, you can see the anti-PD-L1 effect. And really, it seems, at least at the end of their follow-up, that the DNA is quite low. They didn't necessarily clear S antigen in this case, but it seems like it's more than effective than the other vaccine regimens they use. So there might be some hope here as well. And certainly there's some good data coming from the cancer field related to this as well. Then one last thing that I'd like to point out, just because it's one of those things that, I don't know, I've always thought about it but never done it, and nobody knows anything about the B cells. I put this black box there because that's what we know. It's very little study that's actually ongoing about the B cells. And we know it's important because if you use rituximab in these chronically infected patients, they even develop reactivation. So it doesn't even have to be anti-core positive healthy donors. So I'll just plug that, but no plans. Anyway, so just to summarize then quickly, I think adjuvant therapy has the potential if it's used right, because it's activating these toll-like receptors, and we're altering the immune environment in the liver. And this, in terms of, this facilitates a T cell response and T cell expansion in the liver where we need it. These immunotherapy approaches, this one is, I think these are really interesting because we're using the antigen that's in the patient. So we're personalizing the antigenic repertoire for each individual by giving a general immunomodulatory combination. And if we can stimulate T cell expansion, antiviral cytokine production, I think there's a real benefit there. And then, of course, there's more experimental therapies like the gene therapy, but they're in development, and things are moving in this field and moving fast in the cancer field. And they've been effective in animal models. They appeared effective in our patient therapy, but there's a lot more data that are needed. So I will stop there, and I appreciate everybody's attention.